Hello friends. Today my presentation will be on size reduction topic. Myself, Dr. F.S.Dasankoppa, Associate Professor, Department of Pharmaceutics, Hubballi College of Pharmacy, Hubballi. The contents of the topic are introduction for size reduction process, objective of size reduction, advantages and disadvantages. And in today's session, I'll also be highlighting stress versus strain relationship, laws governing size reduction process, that is 3 laws, Kick's law, Rittinger's law and Bond's law. And for the today's session, I'll be introducing the topics and objectives, advantages and disadvantages. The stress and the strain relationship and the laws governing size reduction will be dealt in the next presentations. So coming to the introduction of the topic, as size reduction, the name itself indicates that there is a size. This process leads to reducing the size of the particles. And size reduction process is also termed as comminution, diminution, or pulverization. And as a unit operation, since it's one of the major unit operation in the pharma industry, as a unit operation, we can say that size reduction is a process resulting the large solid molecules into the smaller units. We all know that we call it as a unit operation because there's a physical transformation taking place. The transformation is reduction in the particle size. If the solids are reduced by mechanical means, meaning the powders, the large coarse powders are changed to fine by a mechanical means, then we call this as a milling process. The main objective of size reduction process is to change the polydispersed particles to monodispersed particles. Size range of the particle is it is one of the ideal characteristics for the pharmaceutical processing and designing the various dosage form. In the present slide, you can see the ideal properties. This table shown here represents the ideal property of dosage form with respect to its particle size range. The ideal particle size is a very important criteria for a dosage form. Like for example, if you want the drug to be administered by the lymphatic route, the particle size should range from 10 to 50 nanometer. And for the long circulating carriers which are targeted to the brain or tumor cells, the particle size required is 50 to 200 nanometer. For transdermal use, it is 10 to 600 nanometer. For intravenous and intramuscular route of administration, it is 20 to 2000 nanometer. For ocular drug delivery, it is 100 to 3000 nanometer. For aerosols, it is 1 to 10 micrometer, and for nasal, it is 8 to 20 micrometer. So in this table, we'll come to know that particle size plays a very important role in designing a stable and a therapeutically efficient dosage forms. This slide represents here the pharmaceutical dosage form consisting the particle size range right from 0.01, that is the diagnostic agents and lymphatic drug delivery system, to the granules, which ranges to 1000 micrometer. And I like to highlight two things over here. For oral dispersible tablet, we require a particle size range right from 50 to 600 microns, whereas for the aerosols, it is 5 to 25 microns. Therefore, while designing any dosage form, particle size consideration is very important. To obtain the desired particle size, we need to have an efficient size reduction process. Size reduction process is majorly divided into two major categories. If the material is solid in nature, then this process is called as drying or cutting, and if the material is liquid in nature, we call it as emulsification or atomization. The main objectives of size reduction process are to increase the surface area, which enhances the absorption as well as the bioavailability. The rate of drug absorption is enhanced by size reducing the particles. The particles in narrow size range can be obtained. Like for example, we want for aerosol 5 to 25 micrometer particle size range only. And by this process of size reduction, we can obtain a narrow size range particles. Mixing of the particles with narrow size range is very easier. Hence, the main objective is we want to develop a stable and therapeutic efficient dosage form can be designed by controlling the particle size. There are many advantages of size reduction process also, like there's increase in surface area and dissolution by reducing the size. Example is glycophyllin and progesterone. The content uniformity is maintained. There is enhanced rate of absorption and bioavailability. It facilitates uniform mixing, flowability, and drying process also. It improves the physical stability of the dosage form, especially in case of suspensions, emulsions, and ointments. Effective extraction of the drugs such as galenicals, tinctures, and volatile oils is possible by size reducing the crude drugs. Size reduction process often has major disadvantages also. The major disadvantage is drug degradation, especially to those drugs which undergo hydrolytic oxidative degradation, for example, aspirin, antibiotic, vitamins, or any drugs which are hygroscopic in nature. There is one more thing, that is poor mixing, which may lead from the size reduction process, because as the particle size decreases, there is a charge development on the particle which leads to poor mixing of the product. The next advantage is contamination. Contamination is an inevitable phenomena in size reduction process, because the wearing and tearing of the equipment takes place because the size reduction is taking place by the mechanical means, and this will lead to the contamination of the product. So coming to the next part, that is mechanism of size reduction. We have different mechanism of size reductions, like we have cutting, we have compression, we have impact, we have attrition, and we have a combination of mechanism wherein certain mills are using it, like impact and attrition. So one or combination of one or two mechanisms the mills will be using for size reduction process. Now for cutting, majorly for cutting, especially for in case of crude drugs, leaves, barks, this mechanism is used. We actually use scissor, shear, or cutter mill, and the particle size here approximately obtained is 100 to 80,000 microns. We can undergo a process of compression also, wherein the substance is wet or it contains large amount of moisture in it. You can use a roller mill, you can use a pestle or a mortar or edge runner, end runner mill. The particle size obtained in this case is 50 to 10,000 microns. And by using an impact mechanism, impact is a mechanism where it resembles to just like taking a hammer and hitting it to a solid, wherein the solid disintegrates into pieces. Okay, so this mechanism is seen in hammer mill or disintegrator mill. The particle size obtained here is 50 to 8,000 microns. Next is attrition. Attrition is a very important phenomena wherein the large particles collide with the small particles and the exchange of kinetic energy takes place, and this exchange of kinetic energy leads to the size reduction process. We have colloidal mill in this range, we have fluid energy mill and roller mill. The particle size obtained in this case is 1 to 50 microns. The last mechanism what we are studying is impact and attrition. This is a combination of impact and attrition, wherein this mechanism used by ball mill and fluid energy mill for the size reduction process. So we obtain a size range right from 1 micron to 2000 micron. So depending on the type of the mill and the type of the nature or the nature of the material to be size reduced, the mills are chosen, and it also depends on what type of approximate particle size is required at the end. So my next presentation will be on the stress versus strain relationship, and also I'll be introducing to the laws governing the size reduction process. Thank you.